Ever feel like fungi and parasites all blur together right when you need them most on the MCAT? You're not alone. These topics can seem random, but the test loves the patterns behind them. In this video, we'll break down the key structures, life cycles, and high-yield clinical clues that show up again and again. By the end, you'll be able to quickly recognize what you're dealing with and turn those tricky microbiology passages into easy points. Fungi are eukaryotic organisms with chitin cell walls that can exist as yeasts, molds, or dimorphic forms. This means they have membrane-bound organelles like a nucleus, which immediately distinguishes them from bacteria on the MCAT. Their cell walls contain chitin rather than peptidoglycan, a key structural difference that is often tested in comparison questions. Morphologically, fungi can appear as unicellular yeasts, which reproduce by budding, or as multicellular molds, which form long filamentous structures called hyphae that intertwine into a mycelium. A particularly high-yield category is dimorphic fungi, which can switch between yeast and mold forms depending on temperature, commonly existing as molds in the environment and yeasts in the human body. A classic mnemonic is mold in the cold, yeast in the heat. This temperature-dependent shift is frequently tested in infectious disease questions. Clinically, fungi are often opportunistic pathogens, especially in immunocompromised individuals, linking this topic to immune system function. The MCAT may also test antifungal targets, emphasizing that drugs often disrupt ergosterol in fungal membranes rather than cholesterol in human cells. Understanding fungal structure and classification allows you to quickly identify how they grow, reproduce, and cause disease, which is essential for interpreting microbiology passages. Fungal reproduction occurs through both asexual and sexual mechanisms involving spores and budding. Asexual reproduction is more common and includes processes like budding in yeasts, where a small daughter cell pinches off from the parent, and spore formation in molds, where specialized structures release spores into the environment. These spores are lightweight and can travel through air, which explains why fungal infections are often acquired via inhalation, a concept frequently tested on the MCAT. Sexual reproduction, while less emphasized clinically, involves the fusion of hyphae from different mating types, followed by meiosis to generate genetic diversity. This process produces sexual spores that may be structurally distinct from asexual ones. The key takeaway for the MCAT is recognizing that spores are the primary reproductive and dispersal units, and they are often the infectious form encountered by humans. This connects to broader microbiology concepts like transmission and life cycles. Additionally, the distinction between budding and spore formation can help you identify whether a question is describing yeast or mold. Understanding these reproductive strategies is crucial for interpreting experimental setups and clinical scenarios involving fungal growth and spread. Parasites are organisms that live on or within a host and include protozoa, helminths, and ectoparasites. This broad category is unified by the idea that the parasite benefits at the expense of the host, a concept tied closely to symbiotic relationships like mutualism and commensalism that are also tested on the MCAT. Protozoa are single-celled eukaryotes such as Plasmodium, which causes malaria and are often transmitted through vectors like mosquitoes. Helminths are multicellular worms, including roundworms, tapeworms, and flukes, and they tend to cause chronic infections by absorbing nutrients from the host. Ectoparasites, such as lice and ticks, live on the surface of the host and are important vectors for disease transmission. A helpful way to organize these is by size and complexity. Protozoa are microscopic and unicellular, while helminths are macroscopic and multicellular. The MCAT frequently tests life cycles and transmission routes, so recognizing whether a parasite requires an intermediate host or vector is crucial. This topic also connects to immunology, as parasitic infections often trigger eosinophil responses and IgE-mediated immunity. By categorizing parasites into these groups, you can more easily predict their behavior, transmission, and clinical effects. Protozoan parasites often have complex life cycles involving multiple hosts and distinct morphological stages. This complexity is a favorite testing point on the MCAT because it requires you to track transitions between forms and hosts. For example, Plasmodium species alternate between a mosquito vector and a human host, undergoing different developmental stages in each. These stages often include forms adapted for transmission, invasion, and replication, such as sporozoites and merozoites in malaria. Understanding these stages helps explain symptoms, such as cyclic fevers that correspond to synchronized red blood cell lysis. Another classic example is Giardia, which alternates between a cyst form that is environmentally resistant and a trophozoite form that is metabolically active in the intestine. A useful mnemonic is cysts are for survival, trophozoites are for action. The MCAT may present experimental or clinical scenarios where you must identify which stage is responsible for infection or pathology. This topic also ties into epidemiology, as interrupting the life cycle at any stage can prevent transmission. Mastering protozoan life cycles helps you reason through questions about infection routes, symptoms, and treatment strategies. Helminths are multicellular parasitic worms that derive nutrients from hosts and often cause chronic infections. Unlike protozoa, helminths are large enough to be seen with the naked eye and include categories like nematodes, cestodes, and trematodes. These organisms typically do not reproduce within the host in the same explosive way as bacteria or viruses, so infections tend to be long-lasting rather than acute. They often absorb nutrients directly through their body surface or specialized structures, which can lead to deficiencies in the host, such as vitamin depletion or anemia. For example, tapeworms can reside in the intestine and siphon nutrients, while flukes may inhabit blood vessels or organs. The immune response to helminths is also distinctive, involving eosinophils and IgE antibodies, which links this topic to hypersensitivity reactions. On the MCAT, you may be asked to distinguish helminths from other pathogens based on size, life cycle, or type of immune response. Another key point is that many helminths have complex life cycles involving intermediate hosts, similar to protozoa, reinforcing the importance of transmission pathways. Recognizing the chronic and nutrient-depleting nature of helminth infections will help you quickly identify them in passage-based questions.